part of the ingredients division of Royal Friesland Cantina, a Dutch dairy cooperative. As Domo, we primarily focus on delivering ingredients and final formulations to the infant nutrition industry. With me today is Maria Powell. Maria is our business support specialist, and I'm very pleased that she's with me here today to deliver this webinar for you. We will start, as stated, with the presentation, and then we will follow this by a question and answer session, where we will address as many of your questions that you have sent in. But before we start, let's get familiar with the functionalities available on the platform. Notice the question tab on the left-hand side of the webinar window. Feel free to use that box at any point throughout the webinar when you wish to submit a question. So don't wait, just send in the question as it comes up. We will answer the questions, and you can review the answers in the Q&A tab just above the slides area. The slides from this presentation and other materials are available for download on the Handouts tab above the slide area. Okay, let's now move on to the presentation, to the featured presentation by Maria. Maria, are you ready?
Thank you, Leonard. Welcome to you all, and thank you for joining us today for our presentation on It's All About Growth, How Hydrolysates Help Infants with Cow's Milk Allergy. My name is Maria Powell, and I look forward to talking with you about this topic today. I would like to start by giving you an idea of what we will cover today in our presentation. First, we will give you an introduction to cow's milk allergy, what it is, the mechanism behind it, and the prevalence in infants. From there, we will discuss about how to reduce the risk of cow's milk allergy and the role of protein hydrolysates here. We will finish up by explaining more about different types of hyperallergenic infant formulas, legislation, and international guidelines regarding reduction of risk of cow's milk allergy in infants. During the presentation, we also have a couple of polling questions we would like to ask you to answer, and we will share the results with you later on. So with that said, I would like to start with an introduction of cow's milk allergy. Cow's milk allergy is an immunological reaction whereby the body mistakenly perceives milk protein as harmful. Cow's milk allergy in infancy has a serious impact on both the baby and its mother. Babies who are allergic to cow's milk also suffer from problems like eczema, wheezing, diarrhea, reflux, and extreme vomiting, leaving mothers puzzled and concerned about their health and growth performance. Moving on to the next slide. Cow's milk allergy is often confused with lactose intolerance. In this illustration, you see that hypersensitivity to food can be non-allergic hypersensitivity and allergic hypersensitivity. In the non-allergic type, also called intolerance, the immune system is not involved. Lactose intolerance is an example of this type of hypersensitivity, which is due to lack of the lactose-degrading enzyme lactase. The allergic hypersensitivity, also called food allergy, can be divided into immunoglobulin E-mediated and non-IgE-mediated food allergy. Cow's milk allergy can be IgE-mediated or non-IgE-mediated, whereby it has been shown that IgE-mediated reactions are more common in infancy and non-IgE-mediated reactions dominate in adults. If we take a look at the mechanistic site of the IgE-mediated food allergy, we see the two-step process shown in this picture. After the first contact with the food allergen, the allergen will be attached to antigen-presenting cells, which will in turn present the antigen to T cells. These T cells then stimulate B cells to produce antibodies specific to the antigen, the specific immunoglobulin E, or IgE. These antibodies will be bound to IgE receptors present on mast cells. process is called sensitization. After the next exposure to the food allergen, mast cell-bound IgE recognizes the allergen and gets cross-linked, followed by a release of mediators, which in turn cause allergic reactions. If this allergen is milk protein, for example, beta-lactoglobulin, we call it cow's milk allergy. Moving on to the next slide. The increased risk of developing an allergy has a strong genetic component. Therefore, the easiest and most reliable way of identifying allergy-prone infants is their family history of allergy. The more family members who have a food allergy, the higher the risk of developing cow's milk allergy. But every infant with a positive family history of allergy has an increased risk of allergy. In Europe, 30% of infants have a positive family history and therefore an increased risk of developing cow's milk allergy. Cow's milk allergy is most prevalent in infancy when the immune system is relatively immature and susceptible to sensitization by environmental antigens. Also, the infant's digestive system is not yet fully mature and is still developing during the first year. The stomach pH is relatively high, which is suboptimal for the enzyme pepsin, and the postprandial pepsin output is also reduced. This results in less efficient protein digestion in infants, leading to increased exposure to intact protein in the intestine. Moreover, infants have a higher permeability of the intestinal wall, which may result in a leakage of the not fully digested proteins over the wall into the bloodstream. And since infants have an immature immune system, this higher exposure to large protein fragments will increase the risk of developing cow's milk allergy. The prevalence in early childhood has been reported to be between 2 to 6%, decreasing in adulthood to an incidence of 0.1 to 0.5%. The majority of affected infants acquires tolerance to cow's milk by the age of 5. However, among infants who recover from cow's milk allergy, there remains a strong tendency to develop atopic symptoms such as asthma, hay fever, or reactions to inhalant allergens later in life, the so-called atopic career or atopic march. This emphasizes the importance of reducing the risk of developing cow's milk allergy in early life. Postponing the contact with intact cow's milk protein reduces the risk of developing cow's milk allergy. The first choice is always breast milk, also in allergy prevention. It has a low allergen load and it provides immune active substances to protect the digestive tract. However, in cases where breastfeeding is not an option, cow's milk has been determined to be the best alternative to human breast milk. Still, regular cow's milk formula provides a much higher oral allergen load than breast milk, which increases the risk of developing cow's milk allergy. Therefore, infant formulas based on cow's milk hydrolysase offer a solution as they offer all the necessary proteins, but in a pre-digested form. This facilitates the digestion process and reduces the allergic potential.
Thank you, Maria. There's lots more to come, I know, but I just would like to interrupt you here briefly to remind our viewers that they can submit questions at any time during the webinar. Please type in your question in the box on the questions tab, and you'll find that on the left-hand side in your webinar panel. Okay? Back to you, Maria.
Thank you, Leonard. Going on to the next slide. Hydrolysates are proteins digested into smaller fragments, peptides, and its sole building blocks, the amino acids. So, a hydrolysate is a blend of different peptides and different peptide lengths. If we look at the hydrolysis process, we start with intact protein, for instance, whey or casein, which is digested by enzymes into smaller fragments. The hydrolysis process influences the peptide size. Depending on the level of hydrolysis, we are able to obtain mildly hydrolyzed, partially hydrolyzed, or extensively hydrolyzed proteins. Moreover, the peptides can be filtered in order to obtain a mixture with very small peptides and no larger fragments. This hydrolysis process reduces the allergenic potential of the protein. In milk proteins, many epitopes are present, which are specific surface features on a protein and can be recognized by the immune system. As already discussed, the binding of an epitope by two IgE antibodies is required for IgE cross-linking, resulting in the release of inflammatory mediators like histamine, which are responsible for allergic reactions. Enzymatic hydrolysis destroys both linear and structural epitopes of proteins and is the most effective mode to reduce the allergenicity of a protein. Once a hydrolysate has been developed, several characteristics are analyzed, including molecular weight distribution, peptide fingerprinting, SDS page, amino acid profile, analysis of the degree of hydrolysis, and sensory testing. In allergenicity analysis, we use biochemical methods like the ELISA, in vitro methods like the mast cell degranulation test, and in vivo methods to evaluate the sensitizing capacity of the hydrolysate. For the in vivo safety studies, we collaborate with TNO and the Utrecht Center for Food Allergy in the Netherlands. Clinical trials in infants are performed together with our customers using the complete hydrolysate-based end formula. As regards hypoallergenic infant formulas, the EU directive on infant and follow-on formulas require that the infant formula does not sensitize animals to the intact protein from which it is manufactured. Until now, the guinea pig test served as the golden standard. In addition to that, we are currently collaborating with the Utrecht Center for Food Allergy in the Netherlands, developing a new, more sophisticated mouse model which allows for the measurement of more immunological parameters. In this model, mice are orally sensitized in combination with an adjuvant. After 4 weeks of sensitization, the mice are challenged in the ear, and the acute skin response, body temperature, and anaphylactic shock score serve as a measure of allergenicity of the hydrolysate. The mice are also orally challenged, after which the protein-specific antibodies and mast cell degranulation are measured.
Thank you, Maria. This is a good point to add an interactive aspect to this webinar, this presentation, by means of a polling question. We would like your response to our following question. What is your biggest concern when formulating with hydrolysates? The stability of the formulation, taste aspects, crudeness of the composition, claims, or any other thing you can think of. Please let us know your opinion by clicking on the button of your choice inside the slide area. We'll take a few moments now so that you can respond to the question, and right after the presentation we will provide you with the answers that were given on this question. Okay, thank you very much for your response. Maria, please take it on from here again.
Thank you, Leonard, for this interesting question. I look forward to hearing your feedback later on. So, let's now talk about the market of hypoallergenic infant formulas. When we look at this market, we see a continuous growth of hypoallergenic infant formulas. And within this hypoallergenic formula segment, we can distinguish two types. One is aimed at primary prevention in children at risk to develop cow's milk allergy, and the other at secondary prevention in children who are already suffering from cow's milk allergy. If we look to the protein ingredient of these formulas, we see that the intact protein present in standard formulas has been replaced by either partially or extensively hydrolyzed proteins. We also find this distinction in the relevant legislation. The EU directive on infant formulas and follow-on formulas includes the following requirements for making claims with regard to reducing the risk of allergy to milk proteins. For formulas intended for infants at risk to develop cow's milk allergy, the following is required. The amount of immunoactive protein should be less than 1% of the protein in the formula, and the infant formula must not sensitize animals to the intact protein from which it is manufactured. Moreover, the label shall indicate that the product must not be consumed by infants already allergic to the intact protein. This disclaimer is not required if the formula is tolerated by infants, as proven in a clinical study. If all three requirements are met, the formula is considered suitable for infants who already are suffering from cow's milk allergy. So in the European legislation, we also see the division between products for primary and secondary prevention. And basically, this refers back to the allergy mechanism which we discussed at the beginning of this presentation. For at-risk infants, the focus is on primary prevention, which is targeted at the very beginning to prevent sensitization to the intact protein. For already sensitized allergic infants, the focus is on secondary prevention, to prevent the specific antibodies which have been already developed from recognizing antigens. That brings us to the choice of which hydrolysate should be chosen for which type of hypoallergenic formula. If we look at the literature, we see that partially hydrolyzed protein is effective in reducing the risk of developing cow's milk allergy in at-risk infants. Some bigger protein fractions may also be present in these formulas. Partially hydrolyzed protein may even be beneficial over extensively hydrolyzed proteins, since the peptides are small enough not to induce an allergic reaction, but big enough to induce tolerance to the intact protein. This can be seen as a kind of education of the immune system. Moreover, these partially hydrolyzed proteins taste much better compared to extensively hydrolyzed proteins. Baumgartner et al. Performed a meta-analysis of prevention studies and found that partially hydrolyzed protein-based formula reduced the number of symptomatic infants compared to cow's milk-based formula over a 5-year period, and showed that this effect was comparable to that of breast milk. Therefore, partially hydrolyzed protein has shown to be effective in reducing the risk of developing cow's milk allergy in at-risk infants. However, for already allergic infants, extensively hydrolyzed protein is required, and all reactive protein parts must be destroyed. Successful studies have been performed with formulas using these extensive hydrolyzed proteins, such as Nutramigen, Friso Allergy Care, Similac Alimentum, and Nutrilon peptide. And what are the international guidelines on partially versus extensively hydrolyzed proteins? All guidelines recommend exclusively breastfeeding for at least 4 to 6 months. When exclusive breastfeeding is not an option, they all recommend cow's milk hydrolysate-based formulas for the prevention of developing cow's milk allergy. For infants with a family history of allergies, partially or extensively hydrolyzed protein-based formulas with proven reduced allergenicity are recommended. For infants already suffering from cow's milk allergy, the guidelines advise to use formulas based on extensively hydrolyzed cow's milk protein or free amino acids with proven effectiveness in cow's milk allergic infants. At this time, I would like to hand you back to Leonard for one last polling question.
Thank you, Maria. We would like to ask you another question, and we appreciate very much you choosing a response from the following options. How do you see the outlook for the use of hydrolysase? Very good. There's a growing need for these products. You see it as positive, but the need will remain fairly stable. There will be other solutions for managing cow's milk allergy, and they will prevail, or you don't know how this will exactly develop. We will take a few moments here to allow you time to respond to this question, and we will share the response with you right after the presentation is finished. Okay, thank you very much for your response. Maria?
Thank you, Leonard. So, to summarize this presentation, cow's milk protein hydrolysase are very effective protein ingredients used in hyperallergenic infant formulas. They offer a solution in reducing the risk of cow's milk allergy in infants at risk of developing cow's milk allergy and in infants already suffering from cow's milk allergy. We at Friesen Campina Domo are continuously broadening our expertise and improving our product range. As already mentioned, we are collaborating with the Utrecht Centre for Food Allergy in developing a mouse model as a more sophisticated sensitizing model to guarantee the safety of our hyperallergenic protein hydrolysase. This model can also be used to evaluate the hydrolysase capacity to induce oral tolerance. Moreover, our technologists are currently using the most state-of-the-art techniques for in-depth characterization of our hydrolysase to enable us to steer hydrolysis parameters and to improve their taste. In addition, we are exploring non-dairy solutions for hyperallergenic infant formulas, and we are studying the effects of hydrolysase on protein digestibility. So, this is the end of our presentation on how hydrolysase help infants with cow's milk allergy, and we will now be available for your questions. We sincerely appreciate your time with us today and hope you can visit our website in the near future, www.hydrolysase.com. I will now hand you back to Leonard for the results of the polling question and our question and answer session. Thank you.
Thank you very much, Maria, for these important insights on this topic. This topic is very important for infants in their first stage in life. It's where it all starts, and a good start in life will benefit everybody in general. Before we will go on to addressing the questions that you have submitted to us, I would like to introduce three more of my colleagues that have joined us because we need all the support in answering as many questions that we can possibly get. With me here today also is Marjan Gos. She's our Manager of Nutritional and Regulatory Affairs. Mr. Rick De Waard, he's our Growth Initiatives Manager for Hydrolysase, and Shalonda Hoescher, our Marketing Communications Manager. They will assist Maria and myself in answering the questions that came in through the website. But before we go on to the answers, let's first see how you did on the polling questions. Moving down to the first polling question, to remind you, the question was about what is your biggest concern when formulating with hydrolysase, and the result of that, what you gave us back was the vast majority of you pointed out that taste was by far the big issue when formulating with hydrolysase. Thank you very much for your feedback on this question. Then we also had a second polling question. That second polling question was, how do you see the outlook for the use of hydrolysase? That gave a more spread in the answers. About 55% of you saw there was a very good outlook for hydrolyzate. There's a growing need for these products. About 11% saw it as positive. Very few saw or gave us back that other solutions will be the majority of choice, so it's not very likely. And there's still quite a few that don't know that much about it. Answers with I don't really know. Thank you very much also for this feedback. That's really great to have. So let's now look. As I said, there's quite a few answers or questions that came in. Go for the first question. Question that came in, Maria, maybe you can address this one, is what are protein hydrolysates and how are they produced?
Yeah, sure, Leonard. Well, hydrolysates are proteins digested into smaller fragments, peptides, and their sole building blocks, the amino acids. So basically, a hydrolysate is a blend of different peptides and different peptide lengths. In the hydrolysis process, we start with the intact protein, for example, whey or casein, which is digested by enzymes into smaller fragments. The hydrolysis process influences the peptide size. Depending on the level of hydrolysis, we are able to obtain mildly, partially, or extensively hydrolyzed proteins. In addition, the peptides can be filtered in order to obtain a mixture with very small peptides and no larger fragments.
Okay, that's clear. So it's broken down protein so that the body no longer reacts allergic to it. And as I understand you correctly, Maria, there's quite a variety that we can achieve in different types of products. Then maybe a question for Rick, our growth initiatives manager. This question is more on the use side of hydrolysates. Rick, can you elaborate on the type of hypoallergenic formulas that you see in the market?
Yeah, certainly can, Leonard. What we see is that within the hypoallergenic formula segment, we can basically distinguish two types. One is aimed at primary prevention in children who are at risk to develop a cow milk allergy, and one is at secondary prevention in children who are already suffering from cow milk allergy. So if we look to the protein ingredients of these formulas, we see that the intact protein present in standard formula is being replaced by either partially or extensively hydrolyzed proteins.
Okay, so there's various categories, types of formulations. That's great.
Yeah.
Then a question for Marjan, our Manager of Regulatory and Nutrition Affairs, because these types of different products in the market. What type of hydrolysis do I need for a hypoallergenic infant formula?
The choice of which hydrolysate should be chosen.
The choice of which hydrolysate should be chosen is dependent on which type of hypoallergenic formula is produced. So partially hydrolyzed protein has been shown to be effective in reducing the risk of developing cow's milk allergy in at-risk infants. Some bigger protein fractions may also be present in these formulas. Partially hydrolyzed protein may even be beneficial over extensively hydrolyzed proteins for these at-risk infants, since the peptides are small enough not to induce an allergic reaction, but big enough to induce tolerance to the intact protein. This can be seen as a kind of education of the immune system. And moreover, these partially hydrolyzed proteins taste much better compared to extensively hydrolyzed proteins. So partially hydrolyzed protein has shown to be effective in reducing the risk reduction of developing cow's milk allergy in at-risk infants. For already allergic infants, however, extensively hydrolyzed protein is required, and all reactive protein parts must be destroyed.
Okay, thank you for that elaborate answer, Marjan. Maybe continuing on that answer, a question came in from the other side of the world. If I have to
it looks like. And that's with regards to, it has to do also with, okay, how then do you determine, Maria explained a little bit about animal models, on how to prove hypoallergenicity of glyphosate. Question came in from Sasha. Why do you think the guinea pig model is no longer the gold standard animal model for HA hydrolysates? Why do you use, or why do we look at a mouse model? Maybe you can elaborate a bit on that.
Yeah, so first of all, indeed, for hypoallergenic infant formulas, the European legislation requires an animal study showing that the hydrolysate-based formula does not sensitize animals to the original protein. And actually, no validated animal models are currently available to assess residual sensitizing capacity. But guinea pig assays are frequently used and serve as the golden standard. Oral guinea pig sensitization assays are quite sensitive and have proven value in studies on differences in sensitizing properties of classical infant formulas as well as formulas based on modified protein products. But next to the guinea pig model, we are currently collaborating with the Utrecht Center for Food Allergy in the development of a new, more sophisticated mouse model. One of the advantages of this new model is that the antibody response is IgE-mediated, like in humans. Well, in the historically used guinea pig, the response is IgG1-mediated. And moreover, the new mouse model allows us for measurement of more and objective measurable immunological parameters than anaphylactic shock scores only, which is quite a subjective measure.
Okay. Hope that explains to Sasha a bit about the background on why we're looking at a mouse model and the guinea pig model. Already addressed in the polling question, you gave us back that a property of hydrolysates is taste, and that's an important property. To address that, a question came in, and maybe Maria, you can address this one. Why is the taste of hydrolysates often not as good as intact protein?
Well, as most people know, hydrolysates tend to be very bitter. This is especially due to hydrophobic peptides. Those peptides can interact with bitter receptors in the mouth. And casein can form very bitter peptides, more so than whey proteins. In casein, there are hydrophobic and hydrophilic regions. Therefore, hydrolysis of casein can result in very hydrophobic fragments. In whey proteins, the hydrophilic amino acids are located on the outside of the molecule and are initially digested. Moreover, whey proteins don't have clear separated hydrophilic and hydrophobic regions as in casein. Therefore, hydrolysis of whey results in less bitter hydrolysates compared to casein hydrolysates. Hydrolysates can also have other off-tastes. A peptone taste is the most well-known taste. The off-taste can be formed during the hydrolysis, but can also be formed during the drying process.
And may I add to that, Maria, that early consumption of bitter-tasting hydrolyzed formulas has been shown to result in higher bitter acceptance later in life. So these experiences set the stage for later food choices and are important in establishing lifelong food habits.
Okay, that's a very interesting, intriguing aspect you point out, Marjan, on top of the issues laid out by Maria. Maybe from my experience, like to add that we have a very long history addressing those issues and manufacturing products with very low bitter taste or no bitter taste, and certainly off taste. History that dates back at 20 years. So on to a question that came in from, I think this is from the Netherlands even.
Rick, you can say something about this. Our mildly hydrolyzed formula also used in infant formula. I think our mildly hydrolyzed hydrolysate used in formula.
Yes, I can answer that. That's a very good question. The scope of today is, of course, on hypoallergenic formulas, but hydrolysates are also used in what we call comfort formulas or easy-to-digest formulas. And those formulas are intended for babies with general, non-specific gastrointestinal problems. So they are often based on these mildly hydrated proteins, the mildly hydrolyzed proteins, in order to facilitate the digestion process. And I think that answers the question. So yes, they are used in the so-called comfort protein-based formulas.
Okay, so in a specific segment again, differentiating from hypoallergenic.
Yeah, absolutely.
Okay, very, very good. A question came in on the legal side. Maybe Marjan, you can, from your background, this one best. A legal definition in the EU for hydrolysates and mildly hydrolyzed proteins.
Well, in the legislation you will not find anything about the degree of hydrolysis to range the hydrolysates in different levels of hydrolysis, like mildly, partially, or extensively. The only thing what you can find is in the Annex 4 of the European Directive on Infant and Follow-on Formula are the requirements for hyperallergenicity. And that's the only ranking or the only thing what has been defined and required for the hydrolysate. And what you will find is that partially hydrolyzed proteins will fulfill often the first two requirements, which is the amount of immunoactive protein should be less than 1% of the protein in the formula, and that the infant formula must not sensitize animals into the intact protein from which it is manufactured, and that you can only obtain the first requirement for formulas intended for allergic infants, and that is the clinical trial which has to be performed in infants with cow's milk allergy.
Okay, thank you very much. That's a clear your answer. Another question for you, maybe Marjan, as it is also I think on an important topic. Why do you see whey hydrolysates versus casein hydrolysis-based formulas, and which one is better?
We generally see casein hydrolysates used in hypoallergenic infant formulas for allergic infants and whey hydrolysates in hypoallergenic formulas for at-risk infants. Historically, hydrolyzed casein was used in hypoallergenic infant formulas since caseins are easier to hydrolyze to non-reactive fragments due to their simpler structure. So the quaternary structure of whey proteins is much more complex than that of casein and includes alpha helixes and beta sheets. And during hydrolysis, the peptide bonds are broken and the protein is split into smaller fragments. And as you can imagine, the more complex protein structure of whey protects the inner structure from hydrolysis, and extensive experience is required to obtain the desired type of hydrolysate. So the first hypoallergenic infant formulas mainly intended for already allergic infants were based on casein hydrolysate. However, progressive insight showed that partially hydrolyzed whey protein is very effective in the reduction of risk of cow's milk allergy in at-risk infants, and maybe even beneficial over extensively hydrolyzed protein in the target group, since the fragments are small enough to induce this sensitization, what I already told you, but big enough to induce the oral tolerance. And hydrolyzed whey has also a much better taste compared to hydrolyzed casein. Although the complex structure of whey protein limited the use of whey protein hydrolysates in hypoallergenic formulas for already allergic infants in the past, improved technology and experience make it nowadays possible to use whey hydrolysate in this segment. Still, we think that this market is smaller than the market of whey hydrolysate-based formulas for at-risk. And also blend of casein and whey hydrolysate can be considered to get rates comparable to those of breast milk.
Okay, Marianne, thank you for your very elaborate question. I think there's many aspects to look at when formulating. I think hereby the open invitation to contact us to get the full insights on that. Maybe a last question for Rick. What do you foresee as next innovation in hydrolysate?
Oh, that's a very challenging question, a very good question, of course, trying to look into the future. The techniques that we use to produce hydrolysate, and then you should think of enzymes and filtrations and different types of raw materials, can also be used to develop bioactive peptides targeted at a specific functionality. And then what we see in the market is metabolic imprinting, immunity, brain development. Those seem to be targets that are important towards the future in the next generation of infant formulas. And in that respect, hydrolysate can play an important role. Another trend that we observe in the market is towards non-dairy protein sources in the hypoallergenic infant formula. For instance, what currently seems to be a trend, for instance, rice protein or rice protein hydrolysate, and they appear to be a promising option. And furthermore, and that's a continuous effort that we are dealing with, to improve taste and unraveling the attributes that impact the taste in both single ingredients and final formulas. So this may result in the use of different enzymes, different process steps, etc. So I think there's quite a future ahead still for hydrolysate, where there will be continuous effort on producing new functionality, different raw material sources, and improving the taste on a continuous basis.
Okay, that sounds great. A lot for in the future as well. That almost concludes the webinar and the question and answer for today. We welcome additional questions, and we will forward the questions that we were not able to address to the booth, so we'll address those questions there. So please join us at the booth right there. And I'd like to thank everyone from the team here for their contributions, and hope to hear from you soon. Thank you very much for listening.